everyone, my name is Michaela. I'm the Business Development Manager here at Unravel Biosciences. Welcome to this overview of the rareSHIFT program. At Unravel, we recognize that drug development has left behind rare disease patients. Over 300 million people worldwide live with rare disease, and of the over 10,000 identified rare disorders, the majority still lack treatments. This is due to the extremely large amount of time and money that it takes to develop a drug. Many rare disease patients are familiar with the unpredictable diagnostic odyssey, with over 60% of patients never receiving a diagnosis. The therapeutic odyssey is even less predictable, with 95% of diagnosed patients still having no approved treatment and needing to rely on symptomatic treatment. But what if there was a way to bypass diagnosis and identify mechanism-based treatments straight from the presentation of the first symptom, making medicine predictable? Here at Unravel, our mission is to transform the paradigm of drug development by clinically de-risking new targets using existing drugs. Our hope is to see all patients having access to safe and effective treatments, no matter how complex or unique their needs, and of course, we really prioritize ethical and transparent drug development. We have a novel business development model here at Unravel. We're a biotechnology company that's focused on novel drug discovery for high unmet need patient disease. However, we also have a sustainable service arm called the rareSHIFT program, which is what I'm presenting to you today. This is a rapid drug repurposing program for rare, ultra-rare, and undiagnosed disease patients anywhere in the world. Before we jump in, here's a quick overview of the rareSHIFT program. First, participants submit self-collected nasal swabs. Then we predict existing drug candidates and more using our BioNAV platform. Next, we have an in vivo platform called SquishyWare used to validate these predictions. And finally, we move to clinical validation. Through rareSHIFT, we offer two platforms. The first is BioNAV, which is used to conduct living molecular twin studies. These studies can be conducted at a personalized or population level. We simulate tens of thousands of molecules on real patient data to predict existing drug candidates, identify disease mechanisms, and discover new drug targets. To create a living molecular twin, we first need data from patients and symptomatically healthy, same-sex, first-degree relative controls. Though any tissue can be used, our IRB includes a non-invasive sampling modality, nasal swabs. RNA-seq data is extracted from these nasal swabs, and this data is run through our computational AI platform, BioNAV. This generates a living molecular twin, which can produce a variety of insights. The BioNAV platform is a systems biology causal network model of health based on 121 million drug gene interactions and 364 million unique network patterns. There are 40,000 molecules within its database, including drugs, nutraceuticals like vitamins and supplements, exploratory molecules, and more. You can think of this platform to be like Google Maps. If the disease state is location A and the healthy control state is location B, BioNAV is creating a list of different ways to get from point A to point B using the drugs available. This platform has been validated in a variety of studies, including predicting the standard of care for tuberous sclerosis complex and understanding the non-obvious drug effects of statins to treat COVID-19. Pause to read more and check out our publications at www.unravel.bio/science. We choose to work with RNA because it is dynamic, not static like DNA. The expression of RNA is influenced by external factors like diet, microbiome, medications, environment, activity, circadian rhythm, and more. These changes in RNA expression reflect the true presentation of the disease state. We often get asked about our non-invasive sampling modality, nasal swabs. These swabs cover over 57,000 coding and non-coding transcripts, as well as thoroughly cover neuronal-related pathways as well, with all 1,600 neuronal and synaptic pathways captured. Nasal tissue hosts a variety of cells, which can be seen in the cell deconvolution of our nasal swab samples on the right of the screen. The greatest advantage of our nasal swabs in the rare disease community is the accessibility that they provide. We can sample patients across the globe, and you can see where we've been privileged to work with patients so far in the map on the right. As of Rare Disease Day 2026, Unravel has a disorder portfolio of over 100 disorders, with programs at all stages of the drug development pipeline, from our living molecular twin studies to preclinical models to the clinic, including clinical trials and observational studies. Living molecular twin studies have two common packages. The first is a personalized study. This asks the question, how do we treat this specific patient? It requires one patient and one symptomatically healthy, same-sex, first-degree relative as the control. Both participants collect 8 samples over the course of 48 hours. The results are delivered after 10 to 12 weeks of Unravel receiving back the swabs. The second study is a population study. It asks the question, how do we treat this group or subgroup of patients? This study requires 20 patients with 20 symptomatically healthy, same-sex, first-degree relatives as controls. Each participant samples at one time point. The results are delivered approximately 4 months after receiving the swabs back from all participants. The impact of each study is different. Personalized studies produce a list of therapeutic efficacy predictions, which are very specific to that patient, accompanied by a report. There's also the option of pursuing longitudinal sampling to monitor disease and treatment progression. Population studies provide a list of therapeutic efficacy predictions for each participant in the patient population. But because each patient is sampled at one time point, there's a low statistical confidence in the results per individual. However, the statistical confidence in the population as a whole is high. The participants can be stratified into therapeutic response groups, determining how many subgroups there are in the patient population that respond to therapeutics similarly. You can see in the heat map below that one disorder population has two subgroups that responded to therapeutics in an opposite manner. We can identify biomarkers for these therapeutic responses to determine IE criteria for clinical trials. Additionally, population studies can align preclinical models with therapeutic subgroups and identify other disorders that respond to therapeutics similarly. Both studies accelerate and de-risk the path to the clinic by starting with patient data. The second platform within the rareSHIFT program is our SquishyWare model. This platform is our in-house in vivo models. They're CRISPR-edited tadpoles used to mimic the patient's disease to validate drug candidates by conducting safety, efficacy, and toxicity screens. We can use SquishyWare to determine a variety of metrics, including morphology, physiology, pathology, social and behavioral patterns, drug responsiveness, and more. I'd now like to introduce you to some patients who we've worked with through the rareSHIFT program. These patients have independently worked with their clinicians to pursue drugs predicted from our platform for off-label use, validating our platform's with clinical impact. On the left of the screen is a male Rett syndrome patient who was in palliative care when we met him. Using BioNAV, we identified vorinostat for his treatment, which is typically an oncology molecule. After using this molecule at a very low dose, the patient saw improved sleep quality, normalized blood metrics, and he regained some motor and cognitive function and awareness, and the sharp progression was noted by his physical therapist. On the right of the screen, you can see a female SMC1A patient who we've worked with. She was left unable to walk after a severe episode of status epilepticus and spent over 100 days in the hospital per year and was also transitioning from palliative care to hospice. This patient's clinician prescribed molecules from our list, and 7 weeks later, the patient was walking independently, having minimal seizures and hospitalization, and was removed from palliative care. This patient has continued to be prescribed molecules from our list over time and continues to improve. Depending on the results of each living molecular twin study, there are a variety of ways that our partners can proceed. As we've already discussed, there is the option to proceed to SquishyWare. However, we share all data back for research use, and thus drug candidates can also be tested on external organizations' animal models. In addition, depending on the results of the study, an observational study may be the most logical next step. While Unravel can neither facilitate nor conduct observational studies, we can assist with their design. Finally, in the past, we have seen our partners pursue independent work with their clinicians after living molecular twin studies. However, please note that Unravel Biosciences produces data for research use only, and the results of living molecular twin studies are not treatment recommendations but scientific data. The best path forward is often determined by the safety profile of the predicted molecules, the commercial value of those molecules, as well as the goals of our partners. In the case that the predicted molecules have the potential for commercial value, we would then explore transitioning from the rareSHIFT discovery platform to a discussion around development. This is a less common result of the rareSHIFT program, but it is certainly possible. Unravel's ultimate goal is to defragment rare disease by finding molecules that work across various disorders, making commercially viable markets for those products. Thus, after discovery through the rareSHIFT program, Unravel will then make a strategic decision with all involved parties as to how to proceed forward through co-development or investment agreements. To review the different stages of the rareSHIFT program and their purpose, the first is to define the disease state of the patient using their RNA. The next is to predict therapeutic mechanisms, targets, and interventions using BioNAV. And finally, to validate the targets using SquishyWare. Our greater hope at Unravel is to develop new therapeutics from those de-risked targets. To see our standard pricing, please visit our website at www.rareshift.org and navigate to the Services page. Feel free to contact us with any questions at contact@unravel.bio. Bye, thanks for watching.